Hi, welcome to Neuroverse, a podcast hosted by Carolina
and Clara, where we discuss all matters from neuroscience to philosophy and beyond.
Today's episode is on biomimetic and bioinspired materials.
So cool.
Yeah, I chose this episode. It's a little bit outside of my field, but it's also a way for me to tap into my biochemistry passion again, since this topic has so much about structure-function relationships.
I also find it so interesting because one of the scientists who has inspired me most to become a female scientist is Neri Oxman, who's a researcher who studies biomaterials and material science and links biomaterials to applications in the world and also design for future cities and products and art as well. And I just love that interdisciplinariness of it all.
Amazing. That's so cool. I think I know her. There's a Netflix documentary about her, right?
Yeah, that's how I came across her. Yeah.
Yeah, super awesome. So biomimetic and bioinspired materials are a field that they're inspired in nature, specifically in the structural and hierarchical organizations. And then they try to recapitulate these properties, and things that are highly relevant are things such as, high mechanical strength, color changing ability, autonomous healing, antimicrobial efficiency, amongst many others. And they study these specific properties by looking at non-covalent interactions such as hydrogen bonding, ionic interactions, hydrophobic effects, as well as network structures, dynamic interactions, self-assembly, and then bio-based polymers. And through these properties, specifically the interactions like non-covalent interactions, is what then allows them to probe into different materials and biology and then see if they can replicate it. And so to start off, the terms bioinspired and biomimetic are similar, but they don't mean the same thing. So the standard industry definition is that biomimetics or biomimicry is a field of research where they recreate or imitate the structure and functions of complicated biological systems in non-living systems using simplified or adapted mechanisms. Whereas bioinspiration is the developmental of novel materials and devices with advantageous properties. So you can think of it as bioinspired, the more umbrella field that encompasses lots of different areas, and then biomimicry, a subfield, because it focuses on imitating as opposed to creating novel and different materials.
I think I get it. No, I was just thinking in terms of like the material versus the product that you're creating. Mm-hmm. So is it more common for like material itself to be made of biological substances, and for the product you're creating to be not necessarily a biological one? Or I guess the alternative you could have, well, I guess would they both involve using biological materials as the substance?
So I think that there's a bit of both. So I think in some products, you may just have biology-based materials, whereas in others, you can also just have inorganic-based materials. I think from what I've seen in the research and also from my own personal opinion, the magic happens where you can mix the two. And this also ties to my admiration of one of my female scientific idols, which is Frances Arnold, where she was able to, using enzyme-directed evolution, to bridge these two organic and inorganic worlds. So I think that a lot of the inspiration, even with the inorganic products, does come from the biology. So the way that like spider silks, the way that the proteins in spider silk are arranged, copying that into inorganic materials for building. But also, in more like medicinal and biological applications, oftentimes it does have to be bio-based in order for it to be compatible with our bodies, but potentially also have some element of inorganic composition, such as, for example, like a drug delivery system that has a pH compatibility with our own, but then potentially on the inside, it has an inorganic component. I'm not entirely sure, but this is what I perceive from the literature.
Yeah, I think that's like what I've come across as well. And it's particularly applicable for making things that are used in medicine, like for medical purposes, that are, as you said, biocompatible with our bodies.
Mm-hmm. Yeah. And so in this field, there's a very strong focus on, like I said, network structures, which I think is kind of similar to neuroscience as well. We do place a big emphasis on network structures and dynamic interactions. And, I'd like that the approach of this field is it's primarily nature focused and nature inspired. So nature performs at such a high precision level. From an evolutionary term, there's very few things that are structured and quote unquote designed, if I dare use that term, in such a precise and efficient way. And so some examples that can be touched upon is spider silk, insect resilin, and sea cucumbers. So resilin is the elastic rubber-like protein that's found in insects, and it's specifically useful for wing articulation, movement, and these have desirable tensile strength, resilience, and dynamic stiffness. And these features are relevant at several scales, from the single molecular scale to surface or bulk. So this is like just simply larger compositions and larger scales of the same material, and how it's relevant for things such as drag reduction, wet adhesion, mechanical strength, structural coloration, etc. And then what happens within the biomimetic and bioinspired fields is then it focuses on the molecular building blocks and the end groups of molecular assembly. So within a protein chain, you can have different end groups such as COOH, some amino acid, and these can drastically change the capacity of the protein and then the interactions that it may have. So, for example, for resilin and spider silk, they're both very attractive because, as I've mentioned, they have high tensile strength, they're very elastic, and they're very strong. And yet all of the mimics either can match it with the strength or resilience or extensibility with the elasticity, but it can never quite get it right with all of them. This is carried out through either gene expressions or as well as enzymes that try to then target the specific amino acids or enzymes that are required to produce these proteins, and I guess try to pull and push whatever element of the enzyme or element of the protein that is required to then have this high strength or high elastic property.
So when you say that the mimics don't have all properties, strength and elasticity, why is that so?
So, for example, with the side chains that allow for resilin to be elastic, they try to, well, firstly,
Oh, wait, sorry.
Yeah.
Are you saying that people are trying to combine the features of resilin and silk to make it both strong like resilin and elastic like silk?
I was using them as two separate examples. They don't try to combine the two. It's more like they are scarce products. And so if we want to create our own in order to have more available renewable sources, because these are renewable sources, they're great for the environment. They replace petrochemical based materials and they have all of these amazing properties, but the problem is they're not readily available. And so they're trying to create more. And to create more, they need to look at the properties. They need to be able to produce the proteins that are involved. They need to be able to make sure they get the consistency right. So, for example, they need to add the right lubricant, the molecular lubricants, in order to allow for a higher flexibility without brittleness. And so, for example, this self-lubricating effect of long fatty acid side chains, which allow for elastomers with 93% resilience and a tensile strength of 0.8 megapascals, that's good, but that doesn't reach the 95% resilience and 1 megapascal strength of resilin. And then there's other different approaches. Like you can have a hydrogel approach, you can have an elastomer approach, you can look at the alpha helices and beta sheets. So we spoke about alpha helices and beta sheets on our protein structure episode, which are these secondary structures in proteins that in this specific case, they contribute to the strength and reversible energy storage. What's amazing about resilient and spider silk is that it can stretch without breaking. So this is reversible energy storage. Yeah.
Okay, cool. So there's a lot we still don't know about what makes these materials, and essentially we're trying to make them from scratch at the protein level.
Yes, exactly. So we're either trying to make them from scratch at the protein level, like you said, and this is called rational design. And rational design is when you create something and simply looking at topological architectures of other proteins and looking at the laws and looking at the bonds that are involved and then trying to create something from scratch, which I think is a lot of hard work. Rational design, I think, can also be called de novo design. And then there's another type where they basically use nature as an inspiration. They use that as like the scaffold, as the baseline, and then they build upon it. So, okay, we have this specific protein. Let's, using enzyme-directed evolution, try to push it in this way or that way to get the features that we like about it.
Yeah, very nice.
And then there's adaptive polymeric materials, which, as the name suggests, are adaptive. So something that is amazing about biology is our ability to respond to external stimuli and then alter the physical chemical properties required to address the stimuli and interact with the stimuli. And so this is a goal in biomimetic and bioinspired materials, in creating materials that are adaptive in the same way that our biology is adaptive. And so in order to respond to environmental changes such as pH, temperature, light, ionic strength, mechanical force, etc. And there are so many applications for this, smart drug delivery systems that can either alter the pH, also potentially using some sort of like biosensor that changes color regarding whatever you require it to do, mechanical force, potentially having some sort of device that, depending on the situation, it applies the correct tensile strength or mechanical strength. So this is something that like spider silk does incredibly, when it, depending on its environmental conditions, whether it's wet or in a dry environment, its properties can adapt to the environment. And we can think of, when building buildings and brick and mortar, it would be great potentially in terms of like natural disasters to be able to create a material that can withstand these changes, that can withstand the natural elements, rain, etc. And this would definitely cut back on costs, such as and also reduce pollution.
Yeah, it would be nice to live in a little silk cocoon.
Absolutely, yeah. And another example of a super interesting biological compound is chitosan, which is polyelectrolyte, and it has these reactive functional groups that can form gels, and they're incredibly biocompatible. And as I've mentioned before, with the pH changes, this is exactly what chitosan is used for, in that it is a material that can change its solubility based on the pH of its surrounding. And so this is great for drug delivery, in that depending on different parts of the body where the drug needs to be delivered to, it can change its solubility depending on the pH. Okay. So something else regarding adaptive polymers is the sea cucumbers. So sea cucumbers are cool because they can either stiffen or relax depending on its environment. And they behave in this way depending on the external cues, which are regulated through their nervous system. And this is desirable because they're made up of this collagenous tissue, which is what allows it to have this flexible property, in that it can relax or contract and stiffen depending on the environment. And so this has led to a lot of inspiration of different material uses. And for example, there was a study where the goal was to create these quote unquote living materials, because if you have an organism that can move in a certain way and not needing to constantly encode for its function, like constantly giving it instructions, it just behaves in the natural way that our biology does, then it could allow for a lot of interesting things. So if we take this principle of animals move, and in our case, we have muscles that convert chemical energy to reversible energy, what they did is they created this robot that contained myocytes. And these myocytes came from rat heart tissue. And they then modified it to be responsive to light. And then they ended up creating this 1/10 of a scale version of a ray fish that had a gold skeleton lined with these myocytes that were also responsive to light. And then they were able to control it using light. And it was just this really cool, it produced these really cool undulatory movements.
I remember learning about that in one of my undergrad lectures, and it was cool because we were learning it in a completely different context of like optogenetics, basically, which is a tool that is largely used in neuroscience, and it integrates it. Also, that's kind of related to biomaterials because it integrates a opsin, which is taken from algae. They were cloned from algae initially, and they are ion channels that are responsive to light. And so you can use them, you can express them in certain cell types and use them to, in response to light, elicit chemical changes that result in electrical changes, right, to activate neurons. So I assume that's what they did in that study. And I think that's a really cool application for how you can use like a random protein that was found in algae as a research tool.
Yes, absolutely. And it's super cool that they were able to create this robot that can move and respond to these stimuli, because I think, as I mentioned, this was like a major goal in biomimetic and bioinspired materials, to be able to produce something that's adaptable to the environment in the same way that our biology is. And this is just one example. I'm sure there's many more that I don't know of that show this and how they were able to achieve it.
Yeah.
And on the same topic, I think it's really interesting, this focus that the biomaterials research field has in terms of wanting to program and encode information in the materials. And I think this is quite similar to the desire as for computational neuroscientists and neuroscientists alike to try to decipher how a neuron or a network encodes information.
Yeah, this will definitely relate to an upcoming episode on storing data in DNA, which we'll record next.
Exciting.
And yeah, I think it's interesting to hear that as a neuroscientist, because I know how little is known about how information is encoded, at least in the brain. I guess we know it more for genes, though. Like, we understand the genetic code quite well, having a fully sequenced genome. But I think it would be a really useful thing if we could figure out how materials encode information and how we could use it to store information.
Yeah, absolutely. And as you were saying, it's interesting how for neuroscientists, we feel like there's still so little that we know, but clearly this desire to understand how information is transmitted from point A to point B is prevalent across all fields, including this one, because apparently in all of the fields, they can encode information on a one-way sense. So they can encode the information more or less from the material to the biology. But it's not bidirectional, and they want it to be more bidirectional, and not only bidirectional, but I guess being able to make inference or being able to respond to the stimuli appropriately. Which also reminds me of AI and machine learning and the desire to make it learn in a way that is comparable to the way humans learn, or in a very different way, but this element of learning and this element of like independence and being able to respond to information appropriately.
Yeah, and then in terms of neuroscience and biological engineering, I guess you could call it, what's at the forefront of that is creating brains in a dish, which are called organoids. And essentially it's just putting cells together, putting a bunch of different signaling molecules and chemicals and optimizing that to grow a brain. But like a major limitation of that is you don't have the ability for it to interact with its surroundings. Like you don't have the ability for it to be influenced by sensory activity. Sensory stimuli, because it doesn't have the systems in place to receive that information, like eyes and ears and other sensory organs. And obviously it can't really exhibit behavior.
Yeah.
But then you mentioned AI, which is, it can exhibit behavior, but it's not really able to, well, it's not biological substance, you know?
Yeah.
So this actually then goes into like, it touches on the whole philosophy of mind, like how related is the behavior and the substance.
Yeah, it's interesting. And I like that in this field of biomaterials, there's a very clear structure function relationship. You know, when it goes to studying resilin and all of these amazing proteins that have incredible structures that can allow us to then implement in various different fields and solve lots of different problems. You know, once they can figure out what they know the function, but they haven't quite figured out the structure, and they use so many different techniques to figure it out, you know, electron microscopy and so many more. And I think that for me, this has made me also appreciate a bit more our imaging techniques and how we do strongly rely on them to have an understanding of what protein looks like and what fits into where. Because yes, we can understand the dynamics, we can know how many hydrogen bonds there are, how many ionic bonds there are, but then I feel like we are a bit blind if we don't know where they are aligned to. Like we can know the power of something, we can know the dynamics of something, but if we don't know exactly how they are structured, then we are incredibly limited.
Yeah, so I just wanted to mention scales of structure, because something I came across and that also really interests me is 3D printing biomaterials. And again, I first got introduced to this from Neri Oxman's group, because they have done a lot of work in 3D printing biomaterials such as cellulose, chitin, and pectin, which are all plant-derived materials, and they're really abundant natural materials on Earth. So instead of what you were speaking of, making materials from scratch, they were using these natural materials and trying to fabricate them into structures that were as small as millimeters long to as big as meters long. And they created really these giant structures. But not only that, but others looking at other studies as well, it's actually very challenging to 3D print biomaterials because of their variability. Like it's biological, it's not purely physical. So there's actually a lot of variability. And I think it's interesting, the idea that even if you can understand the structure at the micro scale, like the protein interactions or even the chemical interactions that occur between chemical groups, once you're trying to put it together into a larger structure, it may not act as you would expect. So you need to control for several variables, like the speed that you're releasing the material, the degrees of freedom of the 3D printer, the amount of material that is released at a time. And I mean, this is already a problem for normal 3D printing with like artificial materials that people use, but even more challenging with natural materials. But there's so much promise for the use of this, because you could potentially create devices that can be implanted into bodies, and it could fit individual people on a patient-specific basis. And it could also be used for, you know, rapid on-demand fabrication once it's developed properly.
Yeah, amazing. So interesting. And yeah, like really well pointed out that these are biology-based materials, organic materials, and we don't understand exactly all of their structures. And sometimes when we model it or create it and then try to apply it, it doesn't quite work how we expect it to work. But yeah, it's fascinating. And I also read something about using mycelial networks in order to also be able to create building materials and stuff. So yeah, it's amazing. All of these applications, and nature's right there. We just need to investigate it and apply it. Also related to scales, there seems to be this challenge with modeling and simulating biomaterials, and specifically regarding hierarchical material properties. So going from the single cell level, the molecular level, to the fiber interface, mesoscale, and then macro scale, like across all of these different layers and scales, there's different techniques that can be used to investigate this. So one of them is density functional theory, which is more to investigate on the smaller scale, like atoms. And then there's molecular dynamic simulations, which is done for still on the atomic scale, but for longer periods of time. And then coarse grain simulations, which is for larger systems, and then finite element method. And all of these can tackle and model the material either at the atomic level right up to the macro level, and the challenge is then representing across the scales. And this is also a problem with neuroscience, in that like we can record an action potential at the single cell level, and we can record the oscillations from neuropixel recordings, electrode probe recordings. And then there's this challenge with combining the information that we have from the single cell level to the multicellular level to the behavioral level. And clearly in material science, there's the same challenge of having all of these information about the atomic level right up to the macro level.
Yeah. Another thing I came across in terms of biomimicry in neuroscience is what I was speaking about earlier with organoids. So there's still a lot of work being done in terms of optimizing the development of these organoids. And a major component of them, which is thought to also be a major component of general neural development, is the extracellular matrix, which is an intricate network that is composed of macromolecules that are arranged in a specific way and essentially hold together all of the materials of this tissue. So in order to make organoids, the extracellular matrix needs to be replicated, and to do so, studies have used mainly matrigels or purified laminin, and then used stem cells to create these organoids. But there's also other options which are being tested that may be more optimal, like hydrogel beads and such. And without going into too much detail of all the research that's being done, I just came across the fact that there's still so much that needs to be developed in terms of finding the right material that can replicate the extracellular matrix, and how important this is for neural development and the development of organoids.
Yes, it's fascinating. And I think this relates strongly to one of the goals in biomaterials, where it's to have this programmable material. And so like with the extracellular matrix, there's constant feedback through from other cells and the proteins. And there is this goal to create like a synthetic extracellular matrix in order to be able to have this bi-directional transfer of information.
Yeah, that's a good point. Like you need to get all of the dynamics between all the different cell types that you're using to create your thing correct. And I also just wanted to use this example to highlight the fact that the usage of biomimicry is so important for so many different things, like further developing biological models, and that it's a good reminder that the best way to create natural things is to work with nature. We're ultimately always learning from nature and trying to understand what it does as an inspiration to how we can create our own worlds.
Yeah, I think that's a perfect place to end today's episode. We hope you enjoyed it. There will be resources about all of the papers that we've spoken about on the website, neuroversepod.com.
And also, if you enjoyed the episode, feel free to check us out on Twitter and Instagram at neuroverse_pod and share the episode with your friends.
Thanks for listening. We'd like to thank the Biochemical Society for supporting Neuroverse, a podcast about science and philosophy. This episode is part of our series to disseminate research done in biochemistry.